In our next set of lectures, we are going to drill down and look at the complexities of the heart, cardiac physiology. In my opinion, this is one of the more challenging but also supremely interesting topics that you'll encounter this semester. One of the reasons why it's very challenging is because it's very complex, kind of surprisingly complex. But another reason why it's challenging is because the heart is a muscle, and we just finished covering skeletal muscle. So there's a lot of similarities between cardiac muscle and skeletal muscle, but there's also a lot of differences. And where I see students lose points on their exams is largely because they fail to keep those two topics separate. In other words, they'll mix them or conflate them, where maybe something that's unique to the myocardium they'll apply to the skeletal muscle, or vice versa. So as we progress through these lectures, try to be proactive in keeping what's going on in the myocardium separate from what's going on in skeletal muscles. All right, let's get started. In this lecture, we are going to look at the different cell types that you're going to find in the myocardium, and then focus on excitation-contraction coupling events. In other words, how does the electrical signals that propagate across the cell lead to the contraction that we think of with the heartbeat? So let's first start by looking at cell types. If I take a heart and then look at the myocardium and open it up, I'm going to find two different cell types in there. One cell type is going to be a cell type that participates in the contraction. This is what you see on the screen. These are contractile cells, and when they are depolarized, the result is a twitch or a contraction. 99% or more of the cells that you encounter in the heart will be contractile. They will participate in the cardiac cycle and contribute to the heartbeat. But about 1% of the cells do not contract, and rather have an electrical pattern that is really interesting. The electrical pattern has a consistent rhythm to it, and the rhythm is self-generating or auto-generated, and so these are called autorhythmic cells. They have a depolarization and a repolarization pattern that happens on a specific timer. Now that timer can be adjusted by the nervous system, but if the nervous system has no input whatsoever to that timer, there will be a very consistent pattern that emerges. So sometimes these are called the pacemaker cells, because these autorhythmic cells with their timer and their consistent depolarization end up setting the rate of contractions for the rest of the heart, and therefore set the timer for the heartbeat. Is your heart beating at 60 beats per minute or 120 beats per minute? It is your autorhythmic cells that are dictating that. So we'll be getting into autorhythmic cells later. In this lecture, we're going to focus on the contractile cells. But before we take a deeper look at the contractile cells, let's first write this down. Let's look at the contractile cells in more detail. So the contractile cells of the heart share some similarities with skeletal muscle fibers, namely that their contractile units are arranged as sarcomeres. So in this way, both cardiac muscle and skeletal muscle are striated, whereas smooth muscle is different and is not striated. So cardiac muscle has sarcomeres, and therefore they also have thick filaments with myosin, thin filaments with actin, and the contraction is due to the sliding filament model, where the thick filament myosin heads are grabbing on actin and moving themselves along, causing the entire sarcomere to shorten. In addition, Control of those contractions is dictated by troponin and tropomyosin. And when calcium binds to troponin, that moves tropomyosin out of the way, and that exposes the binding spots, and then cross-bridge cycles engage. So all of that is very similar to how a skeletal muscle functions. But here are some differences. Instead of having one very long cell, myocardial cells are short, and they are arranged in tandem, meaning one right after another. And the connection between these cells are called intercalated discs. So I've gone ahead and drawn 2 myocardial cells here, and the intercalated discs are going to be the junction between one myocardial cell and the next myocardial cell. Intercalated discs are rich with gap junctions and desmosomes. And remember, desmosomes are a type of anchoring junction. And in a previous module, we briefly talked about the importance of those anchoring junctions. And if they are weak, then the myocardial cells can actually separate from each other, and the heart would fray and potentially explode, which is fatal. In addition to this unusual arrangement of cells, each myocardial cell has a whole bunch of mitochondria. How much mitochondria? About 30% of its volume is dedicated to mitochondria. So this is kind of an extreme amount of mitochondria. So if we apply our newfound knowledge of skeletal muscle fibers to this piece of information here, we can probably make some inferences about what type of energy or fuel source is used to generate ATP in cardiac cells. What do you think? Well, it's going to be aerobic, right? So whatever the fuel source is, it's aerobic. The only reason why a cell would have that much mitochondria is if it wanted to do a whole bunch of aerobic respiration. So myocardial cells run on aerobic respiration. And this is really important because there's a definite correlation between aerobic respiration and endurance. The more aerobic respiration that is used to fuel a muscle cell, the more endurance it has. As soon as a muscle cell starts using anaerobic metabolism, glycolysis and lactic acid fermentation, we see fatigue setting in very quickly. So therefore, it's really important that these cells get reliable oxygen and use that oxygen to generate ATP reliably. Can you imagine for a moment what the consequence would be if your heart decided to fatigue? In other words, it decided not to generate as much tension as it should. And it's like, yeah, I'm just not going to move that blood. You would die. So your heart can't fatigue. That would be really bad. And therefore, the heart is designed to maintain aerobic respiration as its almost exclusive fuel source for making ATP to fuel the heart. So there's another example of the structure-function relationship where the structure of the myocardial cell fits the function. Okay, it's probably not lost on you that the fact that we have gap junctions here means that electrical signals can move from cell to cell. One skeletal muscle fiber does not really talk to another skeletal muscle fiber in the way that myocardial cells talk to each other through gap junctions. If I want to stimulate a contraction in the elongated skeletal muscle fiber that I just drew here, a motor neuron would start that by sending action potentials down. Here, the action potentials are not coming from a motor neuron. If I want to start a contraction in this cell here, then I'm going to need to start with action potentials that actually move over from the next cell. So the gap junctions allow action potentials to rapidly propagate across cells and then through the gap junction to the next cell. And in this way, chains of myocardial fibers kind of act like one elongated unit. Let's think about this another way. I have here a chain of 10 myocardial cells, and they're all going to be connected by gap junctions. So let me just go ahead and put those in real quick. Gap junctions, by the way, are never really that big, but they are big enough to allow ions to move. So if I have action potentials occurring here, the action potentials are going to immediately get picked up in the next cell, and the next cell, and then the next cell. And so the action potentials kind of move down the length of this chain, much like they'd move down an axon. But what makes this different than an axon is that the action potentials are setting off contractions. So which one of these cells, I'll just put 3 letters here, do you think is going to be the first to contract? Or do they all contract at the same time? This is going to be the first to contract. In other words, this was the first one to have action potential, so it's also going to be the first one to shorten. And then this one will shorten, and then this one will shorten. And it will shorten kind of like an accordion. So unlike a skeletal muscle fiber, cardiac cells in a chain also exhibit kind of an interesting pattern of contraction, where the first ones that are going to experience action potentials are also the first ones to contract. And this is going to be instrumental when we come back and we take a look at the cardiac cycle, or the sequence of events that leads to a heartbeat, in more detail. So we'll revisit this idea later. At this point, let's take a look at what is called excitation-contraction coupling, which is how does the series of action potentials propagating across the plasma membrane of the cardiac cell cause the actual sarcomeres to shorten. And I'm drawing something kind of similar to what I drew in skeletal muscle physiology, but I'm leaving out the T-tubules. Are there T-tubules? Yes, but I'm omitting them because the T-tubules do not interact with the sarcoplasmic reticulum in a direct way, like they do in the skeletal muscle fibers. So you'll notice the sarcoplasmic reticulum still has plenty of calcium, and we pretty much know the end of the story. We know that calcium is going to bind to troponin. That's going to move tropomyosin out of the way. We're going to get cross bridges, and then removal of calcium from the cytosol is what contributes to the relaxation. And cardiac contractions occur as a twitch, just like skeletal muscle contractions do. So, there are some differences, obviously. First of all, there's no motor neuron. So the beginning of our story is actually going to start with action potentials propagating across the plasma membrane. Where did those action potentials come from? They came from the cell next door, through gap junctions. So let's go ahead and make that our number 1. Okay, there is no DHP like there is in a skeletal muscle fiber. Instead, there is a voltage-sensitive calcium-gated channel embedded in the plasma membrane here. So we're going to go ahead and put that in. And this voltage-sensitive gated calcium channel has a name. It is called an L-type calcium channel. So this voltage-sensitive gated channel is going to open as the wave of depolarization sweeps over it. And that's going to let calcium diffuse down its chemical gradient. Well, where is calcium concentrated? It's concentrated in the extracellular fluid. But remember, there's not a whole lot of calcium out there. It's pretty sparse compared to something like sodium. But there is still some calcium, so let's go ahead and put that in. So some of this calcium is going to come trickling in. So let's add that to our story. So this is where things get a little funky. The calcium that's coming into the cytosol from the extracellular fluid can bind to troponin, and in fact, some of it does, but some of it also binds to a gated channel embedded in the sarcoplasmic reticulum. So what type of gated channel might we find in the sarcoplasmic reticulum? Well, a gated channel for calcium, of course. So there's my gated channel for calcium. Notice it has a notch in it, so it's a chemically gated channel. And what opens it? Calcium. Calcium from the extracellular fluid. So it's this really, really funky thing where some of that calcium will actually bind to the gated channel that then lets out a whole bunch more of this calcium from the sarcoplasmic reticulum. So this is completely different than how it works in the skeletal muscle fiber. In the skeletal muscle fiber, the calcium gate is opened by a change in shape of DHP. Here, there is no DHP, and the T-tubules and plasma membrane are not really connected to the sarcoplasmic reticulum in a direct way. Rather, there's a little bit of calcium that comes in from the extracellular fluid, and that triggers the opening of a calcium gate in the sarcoplasmic reticulum. So this is a calcium-gated calcium channel, which is super weird to say and super weird to think about. But let's just go ahead and put it in for now. So this is step 3. All right, so that calcium that's coming from the sarcoplasmic reticulum, it's coming flooding out here. It's going to be our step 4. And that's going to initiate the cross bridges. So when we think about what binds to troponin, about 90% of the calcium that binds to troponin in a cardiac cell comes from the sarcoplasmic reticulum, where about 10% comes from the extracellular fluid directly. Okay, so as stated, the remaining steps are kind of copy and paste from the skeletal muscle fiber in that calcium binds to troponin, tropomyosin moves out of the way, myosin heads bind to actin, cross-bridge cycles ensue, etc. So now we get to our cleanup stage, and what is different here is that we actually have to restore calcium to two different places. So one place that we need to put calcium back, of course, is going to be back into the sarcoplasmic reticulum, and we're going to use our calcium ATPase to do that, just like we do in the skeletal muscle fibers. So we'll make this step 5. But the other thing that we need to do is we need to get some of the calcium back out to the extracellular fluid, at least 10% of the calcium. So I could use a calcium ATPase there, but it's more economical for me to use secondary active transport because I have a strong sodium gradient. So in this case, I'm not going to use a calcium ATPase because there's a cheaper way to do it. And I'm going to use a calcium sodium antiport. So let's go ahead and draw that in. So this is going to let a sodium ion come in exchange for pushing a calcium ion back out. And it's called an antiport simply because they go in opposite directions. So this is a cotransporter. I may use the word cotransporter on the test, but it is a type of secondary active transport. So this is another part of step 5, the cleanup step. So let's go ahead and write this information down. All right, so those are the basic steps of excitation-contraction coupling in the cardiac fiber. Now, as you can see, there are quite a few differences from the steps that occur in the skeletal muscle fiber. So at this point, I'm going to go ahead and put up a table that I use in class, and I would like you to copy this table down and fill in the information that's already shown. And then what you should do is you should try to fill in the blank boxes on your own. So the ones that I've boxed. Now, real quick, I have PM and SR. PM stands for plasma membrane and SR stands for sarcoplasmic reticulum. Okay, try doing this on your own. Pause the video and make sure you try this. I'm going to reveal my answers in just a moment, but again, it's really important that you try this on your own because it helps commit it to your memory. All right, here's my answer key. Go ahead and pause the video and linger here as needed. I want to circle back around and talk about one thing that certainly bugs me and probably bugs you if you followed the story, the excitation-contraction coupling story above. And that is, if calcium is what binds to that gated channel and opens it, which then lets a whole bunch of calcium out, what's to prevent all of this newly released calcium from continually binding to that gated channel and opening it over and over and over again? In other words, it seems like it would set off kind of an infinite loop where calcium keeps opening the gated channel to let more calcium out. And you'd never be able to completely restore all of the calcium. And I don't have a good answer for you, other than it seems to work. So my suspicion is that much like we have an absolute refractory period in the neuron, where once a voltage-sensitive sodium channel is opened, it takes a little while to reset. My guess is that once calcium binds to this gated channel and opens it, the gated channel probably closes. And then cannot be retriggered for probably I'm going to guess about 0.2 seconds. So for some time. That's my suspicion, that it goes through a reset period, a pretty extended reset period where it cannot be triggered. And by the time it can be triggered, all the calcium has been put back in its place. Okay, with that, we're going to conclude this lecture. And in our next lecture, we're going to look at the action potentials that occur in contractile myocardial cells.